All right, everybody, we're gonna come back now, and we're extremely fortunate today to have Dr. Tad Sonstegard, who is coming to us from Acceligen. And so prior to that, though, he has a long history working with USDA ARS, working in the area of SNP chip development. Dr. Sonstegard is a fellow Cyclone with his BS at Iowa State and got his PhD at University of Minnesota. So welcome, and we're excited to hear more about gene editing.
Thank you very much, and I'm very happy to have the opportunity to speak at my first BIF. I'd really have to thank Allison for laying the groundwork on the technology. I didn't incorporate any of that into my talk, so this is just really going to be a follow-on. From the research and development activities that have been going on in academia to trying to commercialize this technology for applications for genetic improvement in food animals. A little bit more about myself, just to let you know, I have been a cattle breeder since 1976 until I dropped out of breeding cattle to go full-time in my PhD. And then I did do a postdoc also at Clay Center. So I've been very much passionate about finding technology for genetic improvement. And once the SNP chip was up and operational and genomic selection systems were being developed by quantitative genetics, moving to my new position at Acceligen was a great opportunity for me to use technology where we've already learned how to read the genome through next-gen sequencing, but to be able to turn around and take those gene editing tools and write back to the genome the traits that we want to introduce into our populations that maybe they don't have. A little bit of background on our company. Recombinetics, founded in 2008, is really a bioventures company. It has 4 subsidiaries, and the entire platform for all of those companies is based upon gene editing in livestock cells. And that's where our freedom to operate is. We have about 35 employees currently, and most of those are actually in the laboratory. Here's a picture of our opening right before COVID hit outside of Eagan, Minnesota, in our new location. We are currently wholly owned by Acceligen, and we are the only subsidiary that has to do with food animals. The other 3 have to do with human health and longevity, and work all in swine models. So since we are the closest to commercialization, we're currently in an investment raise to spin out on our own away from the parent company. Our vision is better animals for a better planet, and I'll talk a little bit about that during my talk. And we believe that we have a lot of leading expertise, not only in design and application of the gene editing tools and using them in different cell types, but also in the genomics and building breeding schemes of how to use this gene edited animals, either to incorporate into nucleus breeding herds, but also for producers like yourself in the beef cattle industry. Our company has a lot of firsts in this technology. We were the first private company to make animals, food animals, going back to 2013 with microinjection of talons into Nelore embryos. It was done in conjunction with Texas A&M. Allison showed a number of the different traits that we've worked on in her presentation, and that included the beta-gal knockout that was done in New Zealand, the polled animals she talked about, Burry and Spotty Guy. We also have exported semen to Australia and created a second set of progeny from Burry and Spotty Guy there. And 2 of the daughters we kept, and they should be calving actually this week. So we'll have the second generation of animals from those proof of concept animals for polled. We've also made 2 different rounds of castration-free pigs. So what does that mean? That the animals stay in a state of pre-puberty and their testicles or their ovaries never develop. The point of that is that in Europe, surgical castration is under a moratorium and they're supposed to stop practicing it. And so this is a way to allow animals to not be castrated and also to avoid boar taint. We've also worked in some disease resistance traits, which I don't show here. Those are still very in early stages of R&D. And so I'm going to talk about a couple of these animals up here, but also some of the ones that we have that are coming down the pipe right now for commercialization. So what is our product really? It's a genetic opportunities for producers all the way from a smallholder up to a large genetic company, where we take the process of gene editing and introducing the traits that you want into your population. And we de-risk that process through our expertise in our breeding platform. So we have our own internal discovery pipeline for different traits. Some of that I'll talk about in an upcoming slide. We do have probably the longest amount of experience relative to even academics in working with CRISPR and talons in livestock cells. We've developed our own pipelines for off-target prediction, and I'll talk a little bit about that because that's one of the main topics for regulators. And most of our applications would be considered non-GMO for gene conversion, and I'll talk a little bit about that. The targets, they vary. We really want to use those traits that the producers want to put into their populations. We don't develop our own applications typically. We go with what the producer wants. Most of those traits are already in the literature or they've already been discovered by us. And then we have our own capability in advanced reproductive technologies. Most of that's in swine, but we do use partners in making those animals. And then our goal here in this breeding platform is to get total acceptance. So what does that mean? We target natural alleles, those that could be introduced into your population by conventional breeding, but you don't want to bring along all the unwanted genome that may be in that other population that's going to disrupt your current genetic program for selection. And we try to match the method that we use for introduction of that trait to the national policy where those animals are going to be sold and eaten. We also incorporate external audit on our quality control of the production of our animals, thereby providing good stewardship before those are released into commercialization. And we also work with breed associations on getting registry, which is of course very important in both dairy and beef. So just outward facing as a way to get better acceptance for the technology among consumer, because at almost every talk I give, whether it be an investment group or a producer, the question is, what about consumer acceptance? Consumers will never accept it. I think there's plenty of information out there and surveys have been done that if you meet the shared values of the consumer, they will accept the technology as it's incorporated if it provides value to them. And so a lot of the traits that we focused on early on have to do either with disease resistance, better functionality of the animals, animal well-being, which includes the polling. And then we also try to address the topics related to use of natural resources by our livestock, so the carbon footprint and also climate change. A lot of times these are gaps that conventional animal breeders didn't consider when they formed the breeds, and now to go back and do crossbreeding to bring some of those traits in, of course, that's disruptive to their programs. These bottom three categories of natural resources, animal well-being, and climate change are all reasons that companies have started up to create fake meat using plant-based materials, or growing cells in a dish that we're going to eat, so laboratory meat. A lot of investment has been in those areas, but truly I don't think they're any more sustainable than the excellent breeds that we've already created. It's just whether or not we can spread those breeds around the globe. And I'll talk about where the opportunity is, especially in the tropics, which has been touched upon as being a region where there's a lot of cattle that are very inefficient at production of animal protein. So more inward facing and what we want to do with our partners who are the producers, it's really adding another component that complements what they're already doing out in the field. So we know that big data and machine learning are going to be very important for phenotyping animals and gathering information that's going to help us with management and selection. A lot of investment is going into this area right now, especially around predicting animal health outcomes and trying to get to those animals before they get sick. Of course, you all know about genomics and genome selection. It's a mainstay right now, especially in the beef industry. And then using that information to drive to find new sequence variants that have major effects on traits, that's where genome editing comes in if you want to move some of those traits from one population to another. A lot of times right now, we think about single gene traits, and those are the ones that are the biggest targets for gene editing, but I'll talk a little bit about doing multiplex gene editing, which is editing more than one location at a time. So really the outcomes of what we want to do is accelerate genetic improvement by complementing current programs, increasing sustainability of production, and really helping meet the challenges of global food security using the genetics that we're comfortable with. Acceptance by regulators. So the third piece of comprehensive acceptance is really acceptance by governments and the regulators that run their programs for determining whether or not that animal or that genetic product can be commercialized. Here I'm showing a map that's produced by Foreign Ag Service at USDA. The green represents those countries where the commercial pathway seems open for gene edited animals. Yellow represents those countries that are still forming frameworks. And then the hatched are places where there's some conflict between what the regulatory processes are. It's quite unclear. You can see a lot of the map is not colored at all. Those countries haven't developed any regulatory frameworks yet. And I will say even those countries that have developed, those first ones just started coming online in 2015. If you compare the systems, no two systems are identical. But there is some agreement that off-target analysis is critical. So what does that mean? If you're using your editing tool in your cell that you're going to turn into an animal, either a clone or an embryo going into a recip, that the DNA is not being cut at some other location you didn't intend it to. They want to know about that. They consider that a risk. So even though there's up there between 30 and 100 de novo mutations that are not caused by editing, it's just part of the natural process of reproduction. And those mutations are random. They don't regulate those. They just want to regulate those genomic alterations that were done while you were trying to do your intentional alteration to make sure you're not putting out an animal that has some sort of a defect or an animal welfare issue. One thing that is clear is that there's starting to be alignment of the Cartagena protocol. So the biotech laws in South America are based upon that, and that's where they come up with their non-GMO policies. So what does that mean? If you're not inserting a foreign DNA and you're just cutting the DNA and allowing it to reanneal naturally, or you're just converting to an allele that exists in nature, you're going to get regulated as non-GMO, so conventional breeding. I think that there's a lot of confusion about what's going on in the US. In part, that's probably related to the agency's inability to communicate very well about what their process is. I think they stay guarded because this process is founded in law. And when you get an approval of a product, I'm sure that there will be some sort of activist action to try and stop it. We've already seen it with Aqua Bounty, who's been in court for a long, long time. However, now they're actually on the market and they've pre-sold out all of their fish online because the retailers refuse to carry it, but it just shows that the consumers accept it because they see it as a sustainable product. Relative to the process with FDA, I think in the past, they certainly were going to regulate everything as a drug. I think that if you could introduce a trait through conventional breeding and you can mimic that through gene editing and it's a naturally occurring variant that we've already eaten safely for many years, they now will consider discretionary enforcement on those types of edits. So what does that mean? It means they will not take it through the drug regulation process, but they have to do an assessment to see whether or not it requires that process. In Australia, there's a type of what we call asynchrony, and that's because they have two regulatory systems. One is based on the gene tech regulators law. And in their revision for gene editing, they say if you just break a gene, it's mutagenesis, there's no regulation. Nobody's tested that yet that I know of on the animal side. If you do an exact conversion of allele to mimic something that's in nature, if you used a little DNA oligo to trick the cell into doing that repair, that's a GMO. On the food side, if it's not a novel foods, that means it's an exact allele that exists in nature. They're not quite sure, but they don't think they're going to regulate it. They haven't really tested the system yet. But if you break a gene and it creates a new protein or a shortened protein, they may have to evaluate it as a food safety risk. So you can see those two things don't make any sense, and they have to fix that problem. For the EU, you can forget it. Sonny Perdue called it the no tech zone. I mean, that's a bit harsh, but EU law mandates that all changes to the genome that were done in the laboratory to a cell are GMO. However, I think that they're very concerned that they're going to fall behind in using this technology for good and improving sustainability, that those things are being reconsidered, but it's going to take a long time there because they're still really stuck in what's called the precautionary principle. So I'm going to summarize our view of what the commercialization process is, and then I'm going to go into some examples to end my talk. So before you start a company or you decide that you're going to start gene editing an animal that you're going to sell or commercialize, you have to get your legal house in order. So that includes, you have to have a license to use those tools. In our case, we have a CRISPR-Cas9 license to be able to make commercial products. And in actuality, there is no research exemption on a tool license. It is a patent infringement. So we'll see how that plays out over time, because there are people using CRISPR-Cas9 to develop their own products, and that could cause some litigation down the line. Certainly, if you discover a trait and you write a patent as to the method for introducing that trait through gene editing, that can be eligible for IP or a patent, or it could be a trade secret. And then there's also the ability of how you introduce that trait into a cell. In our case, we have a lot of method or utility patents related to introducing changes into a livestock cell. So all combined, that gives us the ability to work on the traits that we do and to try and commercialize those. I'm sure somewhere down the line, there'll be litigation between two different groups at some point, but it's hard to see how those things will play out right now. Later on, after you get some animals developed and you have some commercialization potential, of course, you need to have contracts for your partners, and how are you going to collect revenue based upon these genetic opportunities. And if you want to put these animals in the tropics, especially, you have to have international offices, because you have to have a local address in order to get regulatory decisions made on your products in those countries. You can't do it from the US, for example, into Brazil or Argentina. So the second major component is, of course, animal development and deployment. That starts off with your breeding tool design and testing, and of course, trying to predict that you can avoid off targets in your genome and be very precise in your editing. Making that proof of concept animal to show you can change the phenotype through the gene editing, and to show you can also do that exact change without any off targets. And then in some cases, especially if it's something that doesn't exist in nature, you need to phenotype those animals. I'll say that the trait license or IP and these A, B, and C in part 2 are things that government and university research labs have done quite a bit in so far relative to livestock. I know that 2 trait licenses have been given out for disease resistance well, there's actually been 3 that have been licensed out, 2 in pig, 1 in bovine, and 2 of those actual licenses were failures. The traits didn't work. But there is one that is working, and that's, of course, the PRRS-resistant pig CD163 that Genus is working on. To go past that, to really get to commercialization, if you want to get some sort of a decision made to be able to commercialize, you have to have a standardized procedure for making your animals. If you want a ruling on the regulation as a trait, that same procedure has to be used to make every animal. So that means you're basically doing something that's compatible to an ISO 9000, a lot of high standards. If you get an audit, you better be doing it that way. If you're going to bring that animal forward to commercialization, there can't be any mistakes made. And you really need to have a breeding plan with your partner as to how you're going to integrate it into either their nucleus herd, where you have a more vertical program like that in swine, or in a more distributed program like that in beef and sometimes in dairy. How are you going to use those traits in these animals that have been differentiated to best bring those changes through the population? Relative to regulation and policy is the third leg. Of course, you need that government decision to get to the market. I've talked about GMO, non-GMO decisions in Argentina, Australia, and Brazil. There's, of course, the INAD decision for FDA, and then whether or not it's a novel food in Canada and also in Australia and New Zealand. Another important component that we consider, and Donald talked about it earlier, and that is being able to register these animals. It's very important, especially for seed stock producers, to maintain the value. They're not just some rogue animal that has an edit. Nobody's going to use it if it doesn't fit within the registry or the herd book. So we've been working actively in that process. We did several years ago with Holstein and now working with Red Angus Association. You also need to have that registry in order to do import-export to get your CSS code. And you have to follow the IETS rules if you're making embryos. So not only do you have rules for the genetics that are the same conventional regulation we already have to follow, but you also have to consider things like trade. So if you're introducing a trade into a country that exports all their meat, is the terminal market going to accept that meat if it's been gene edited? And then, of course, there's labeling, which we don't know how that's really going to play out, but we have to be ready for it. So now I'm going to switch to the examples, and I've shown 5 different categories here. These are the 5 traits that we believe are commercially ready in livestock. And you can see it's cattle focused, both dairy and beef. The traits we're working on in fish and swine, I'm not going to go over, but we do have an effort in total of 14 different traits. Most of those are related to disease resistance, and they take a much longer time to prove out. So I'll just talk about these because they're the easiest to see is actually being commercialized, and I'll give you a viewpoint of where they're at along the way. I'm going to start off with polled. Allison showed a diagram of what that looks like. That's a non-gene edit. So it's a regulatory element of some kind. It's not a part of a gene. And it affects expression probably of a non-coding RNA that affects expression of another gene. We know what that other gene is because that gene is mutated in polled sheep. If you didn't know, the polled animals also have double eyelashes. And I believe those colored spots that look like where they're supposed to have the horns in our cattle, that's because prior to horns being formed during fetal development, there's actual migration of pigment cells to those spots. Phenotype and food safety, it's a known allele. So we believed it was safe, but Allison did a great job using the scientific process to show that it's no different. So does that need to be done again? I don't know. I think she proved pretty soundly that there's no difference. When you take an allele from nature and you put it in a new genetic background, you still end up with the same meat and the same milk. So the proof of concept animals have been going from 2015 to 2021. And here I'm just showing all the babies that we made by breeding Holsteins that some Japanese buyers didn't want to take back home. So we were able to use those animals in Victoria through a collaboration with TLG, which is a genetics company in Australia. We did receive pre-approval in Argentina for introducing the polled trait into cattle. We have a Holstein cell line that's being sent to Argentina to clone with a company called Chiron. And then once we do the sequencing and that animal's on the ground, we'll be able to get a non-GMO approval there. And this just represents that document. So that's all I'm going to talk about with polled. Certainly, if anybody's interested in that here, we're happy to talk about it. Milk yield QTL. So this one's somewhat a little bit controversial with the science crowd. These are changes in the coding sequence of both growth hormone receptor and DGAT1, which is an enzyme involved in fat metabolism. These were QTL that were found by Michelle George's lab in the early 2000s. They do have an effect on milk yield, and the milk yield allele was derived from the Holstein breed. And so the idea here is that we're going to introduce this into Zebu dairy cattle. The allele frequencies are either 0 or very, very low in the case of DGAT. Again, these are natural alleles, and the phenotypes and food safety are known. We've been drinking milk and eating meat from these animals for years that have these alleles. These alleles are also throughout many of our beef cattle populations. They're just sort of in flux, not under selection. But our goal here is not really for the commercial market to make dairy cattle better, although if they prove out, we certainly will use them in Brazil, but it's really for smallholder impact. Our goal is to double smallholder revenue. So this is just another representation of the amount of greenhouse gas produced by country versus their milk yield. And you can see that in sub-Saharan Africa, that carbon footprint is huge compared to their milk output. So the idea here is we're gonna use this research to help end hunger and increase the revenues of the smallholders. And this is just an interesting editorial that was in Nature that says they looked through all of the research that was geared towards the smallholder, and they could only find like 10 things that were important. And their take-home message is that most academics are worried about their career, not their impact. So we were fortunate enough to get funding from the Bill and Melinda Gates about a year ago in a project where we've proposed to produce 16 founder animals, 8 from the Holstein side, 8 from the Gyr side. Those animals were sampled from Transova's genetic program on Holstein, and also from Baza Fazenda, which is one of the most famous Gyr herds in Brazil. What we're doing is not only are we adding the high milk alleles into the Gyr, but we're also putting in trypanosome tolerance alleles that we believe need to be tested out. And then the Holstein is getting a heat adaptation allele, which I'll talk about later, called SLIC, and also some the trypanosome alleles and some for TB resistance. What's interesting about this project is we're not just making one gene edit, we're making anywhere from 3 to 5, and we're using a different kind of cell that we've never used before, and that's an embryonic stem cell. These stay in culture longer. It allows us more time to introduce the edits. So whether we do it by multiplex or we do it one at a time and then select and re-edit again, it allows us the ability to take these out a long time in culture and still be able to produce calves. And we've actually done our first transfers on the first edits from the Holstein side to make sure that we're able to clone these animals. So we're very positive because the cells are growing quite well. The third one I'm going to talk about is on heavy muscling. So if you remember the first animal we ever made, we introduced changes into the myostatin gene, or GDF8. There's in nature 9 different alleles that cause heavy muscling, and this is because of artificial selection by man. When they see an animal that has more muscling, they're like, oh, I want to use that one to breed. So those alleles are very prevalent in these breeds. And we know that if you debilitate the function of myostatin, you get an extra round of replication of the somite cells before birth, and you get more hypertrophy after birth. And the offal or the carcass yield is higher because the offal is somewhat smaller. So really, this is a carcass trait. We decided, Allison showed this picture earlier, to introduce it into Nalor, which we know has a conformation that some people say is like a giraffe. But they're very tropically adapted and haven't been selected heavily for muscling. So in this way, we're just shortcutting very quickly to moving away from a non-tender beef to a more tender beef, and also a higher carcass yield, which is what they pay for in the market in Brazil. The phenotypes and food safety are very well known. They've been eating this type of animal in Belgium, at least, for years. And 2 weeks ago, we received a regulatory decision in Brazil. We finally did the next-gen sequencing on these animals. We looked for off targets, and what we were able to determine is there were no off-target changes. Here I'm showing Samson from 2016. He's still around at Texas A&M, just much older and very lonely. He's had one offspring there. That is from a Jersey Red Angus cow that Chuck Long put with this bull. But we do have a buyer for the semen, and he's really interested in improving carcass traits in Nalor. He's using a lot of ultrasound, but in this case, he's going to create a new line that has a change in tenderness and also carcass yield that he wants to investigate. So we hope to get that done here this summer and have the first commercialized gene-edited animal that has a sold genetic product. Moving on to coat color. Again, these are alleles that exist in nature. There's more than 300 genes involved in coat color in livestock. In this case, we're looking at ones that are involving black and red coat color. again, alleles that have, we know the phenotypes, we know that the food is safe to eat. So really, what is this opportunity with coat color? Well, the Angus breed is almost 200 years old and it's the #1 breed. They built a fantastic marketing program for their beef, and others have taken on that coat color to try and get within that program. So I'm showing here the Macomb photo from 1824 and a modern Angus bull. But the red came into that Angus breed, we believe, sometime in the 19th century through some sort of admixture event. And in 1954, of course, the Red Angus Association formed, which is near and dear to my heart since I was a junior in that association. And it's now the #3 breed in the US. However, there's some limited genetic diversity because of this admixture event represents a single crossing allele. And so not all Black Angus were carrying this around, this recessive red allele around, just a very small proportion of the population. So we know that the Red Angus breeders have tried to bring in Black Angus genetics by doing black-red carriers, a very long, tedious process that can be expensive because the black-red carrier doesn't have a lot of market value. So is this really a commercial opportunity? When I joined the company in 2015, I thought about it a little bit. And during that time, another red gene popped up from Black Simmental called the Red Charlie gene. So that was another new mutation in MC1R. Still was thinking about it. And then we were approached by a online gambling person from the UK, and he wanted to make the perfect red cow that would mark the Armageddon. So this is covering up my headline, but it basically says apocalypse now if you get the birth of a red heifer in Israel. And so the idea was he wanted to pay us to make this perfect red cow. We're not in the business to gene edit to make religious icons, although we did look into what the edit may be, and we considered the ability to introduce this dominant red mutation that popped up in 1980 in Holstein and was published around 2016. And then also at the same time, being involved in coat color was kind of controversial because there was certainly some news that Mike Moss was going to release an article on how the black coat is inhumane, especially with animals being in the hot sun. So an article really against black coat color. But we were approached by a partner, and their idea was that the red bulls that perform the same as their black founders, if we could clone them and change their coat color and really provide the Red Angus breed with a chance for more diversity and opportunity for genetic improvement. So we started the B2R project. And really the idea is to make the perfect Red Angus replica of a Black Angus animal. And those first calves are going to be due the Q3 of 2021. So really a COVID related project that we've been working on very hard and very quickly. So the idea is you go to your favorite Black Angus bull that you have access to, you get a cell line made by Transova. They do a great job at that. They send it off to our lab. We do the gene editing and the cell screening to find that perfect cell for cloning, send it back to Transova, and then hopefully that red version is going to be born very soon. And it'll be a great commercial success. So some of our process, we have multiple methods for making the red coat color. One of them is even dominant. So if you breed that red animal to black, the calf will be red. We have a perfect pre-cloning selection. The time to puberty to make this animal is no different than normal. You just have a small time in the lab and then you go to the IVF process and it's just like that. And 9 months later, you have your calf. It's easy to detect our edited allele, and we have methods for doing that. No matter what type of edit we make at those loci. And all of these qualify as natural alleles for regulatory assessment in all the countries. And we've already, as you heard from Donald, had approval of both these edits by the Red Angus Association for registration as category 1A. So on to the last trait. Let's talk about slick. The idea here is to expand the use of Angus genetics in the tropics. And I'm showing the tropics as being the Caribbean basin and Brazil, but mainly Brazil, where they have 58 million adapted Nelore cows. And they've been doing straight bred Nelore breeding up until recently. Now they've been importing in Angus to make what's called F1. and the F1 categorized as a certified Angus beef in Brazil. 80% of their beef is eaten domestically, so it's a very popular product. And you can tell that because you look at the semen use. So they're not able to use Angus bulls on the ground. They've tried to develop tick vaccines and methods for keeping them alive through the heat. So they had to go to a fixed time AI program. These programs are massive. And you can see from semen sales here in this graph that we're already up to around 7 million doses being sold in Brazil for Angus semen. That represents about 6% of the Nelore cow population being bred with Angus semen. And we ran some numbers on what that means for sustainability, since an F1 reaches slaughter weight 6 months earlier than a straight bred Nelore. So it represents about a billion kilograms of beef on that 180 days less to market. That means that you save land use the size of the country of El Salvador, about 20,000 Olympic swimming pools full of water saved, and about 350 hours of driving relative to kilos of CO2 emitted. So you can imagine if that you could even increase this better by making the Angus part of the genetics thermal tolerant. The F1s might even do better. So you could get into that semen market and help expand it to higher use, but also there could be an ability here, which we're very certain of, to put Angus bulls on the ground to also do natural service and increase the size of that market. So we're working on that. In 2018, we put Genzelli down on the ground. This is a red Angus calf that had the Senepol allele, homozygous. She was a clone. And here you're just seeing a photo of her. She was a bottle calf because she had to be taken out by C-section. She was born in Brazil. And on the other side, you're just seeing North American genetics at about 85 degrees in the morning, 10 o'clock in the morning, and the calves are already panting and standing in the shadow of the tree. Genzelli preferred to lay in the sun. So the edit worked phenotypically. She was heat adapted. And you can see the short hair phenotype. So where did this gene come from? Oh, the Caribbean basin. When the Spanish and Portuguese dropped off these cattle, a lot of them had to mutate and adapt in order to survive. And those animals that adapted spread on their alleles. And so we've done an extensive survey of all of these Criollo breeds. And I'm just showing some of those here. Most of them come from Colombia, but you can see if you look at their ears, there's no hair in there. They all have the slick phenotype. So when we first looked at Senepol and then we genotyped all these other breeds for the Senepol allele, they didn't have it. So we're like, what's going on? Why are they slick? So we just went to prolactin and we went through and did sequencing of all the prolactin genes from these breeds. And you can see that we found 6 different versions or mutations within the prolactin gene. All of them shorten the gene and make the animals have short hair and heat tolerant. And you can see that none of the alleles except one are specific to a certain breed. So that means they all come from a mix of feral cattle, and then these breeds were formed after those feral cattle were re-domesticated. Farmers have already known, ranchers have already known about this allele. It's being integrated into dairy cattle now by a company called Thermal Regulatory Genetics. And also in Australia, the Senepol was brought in to help build the tropical composites. So it's out there commercially. There's more than 100,000 animals out there that have the slick allele in them in the tropical regions, and it works quite well. So we introduced it by microinjection into embryos, and here I'm showing 2 full sisters, 100% Angus. The one on the left, I think you can tell, is the gene edited one, and the one on the right is normal. So no take. So quite a drastic difference in the hair length. So we had a couple of animals born in 2020. Again, we've already received pre-approval as non-GMO in Argentina, and we've already received approval from the Red Angus Association to register these animals. So they are in the process of being registered, and the bull and the heifer are unrelated. So we'll be making IVF embryos for those for transport down to Brazil, and also collecting semen on the bull. We have entered a pre-review phase with FDA, and that will move to actual review for discretionary enforcement next week. And we'll also be applying for non-GMO status in Brazil so that we can import, export in a conventional method. On top of that, another 20 of these Angus animals will be born here starting in July at the University of Florida. So we believe in sort of wrapping up the opportunities for impact, the biggest ones are in the tropical zone, especially for beef and dairy, and making that impact on greenhouse gas emission and improve production. Slick definitely affects animal health. So the animal's not under heat stress. It doesn't suffer from as many diseases that are common in the tropics, and it's better able to fight off even ticks, we believe. We've shown that there's rapid and sustainable change by gene editing, and this really is an opportunity to replace the inefficiencies of crossbreeding that we use to adapt our high performance breeds. And what's nice about this, it all aligns with the regulatory systems of South America, with the timelines, and the processes are based upon risk, and they support innovation. So it gives us opportunities as a company for investment and business growth. I thank you for your attention. Sorry if I ran a couple minutes over. If you have any questions, you can either talk to me or Sabrina, who's in the audience. And I'm just acknowledging some of our partners in this work, and happy to take any questions if we have time. Single cell embryo is one method. The other is a cell in a dish. It can be, there's multiple types of cells you can clone. There's no method for doing that I know of. So I think the question is, can you edit the germ cells in an adult animal or an animal that's going through puberty? And I don't know of any method like that. So the avatar or the surrogate sires that Allison talked about, they knock out the ability to produce those sperm cells, and then you're supposed to seed them with another sperm cell, but there's multiple methods for do that. We're not working on that technology right now. We'd rather just adapt the genetics to the environment. We can do it through an embryo, but Allison talked about mosaicism, which means the editing's not perfect in an embryo. You can get multiple outcomes that some you may not want. So we actually prefer cloning because it cuts down on generation interval time and you can get to market quicker. We need to generate revenue. So, it takes 2 years still to make genetic product from an animal. So we would prefer to go through cloning.